I'm Jim Speer. I'm a professor of Geography and Geology at Indiana State University. So Brendan Kearns has worked with the DNR before and had an interest in the mussel shells in the Wabash River. And I forget where we talked about it, probably during one of his talks, but we were talking about his interest in mussel shells. And there's an area of dendrochronology or paleo research called sclerochronology, where we can actually look at the layers in mussel shells and any bony structure, and they put on rings just like tree rings do. So we could look at the age of mussels, we could look at the chemistry that's involved, that the mussels are picking up, that the shells are picking up, and look at that back through time with annual resolution. So Brendan and I started talking about that, and we applied for a scientific research permit to get mussels out of the Wabash River, and of course Brendan knew where some good mussel shell locations are along the river. And mussels are protected in Indiana and across the US, so you can't just go out and even get dead mussel shells, and we're collecting all dead organisms here. But you need to have a scientific collection permit to be able to do that. So we were able to work together and look at the science of these, and we're starting to understand a little bit more about the mussel kind of life in the Wabash River. So you have the normal mussel shell, and actually you could see the ridges on the surface, and most of those ridges are probably annual rings, but we need to be able to look at them closer. So we take a cross section of the shell, so we've got the full shell, we take a cross section, and then we mount it on a glass slide, so just like a biological slide, and then we sand it down, polish it very fine, so we can actually transmit light through that slide, and we can see all of the bands, all of the rings in the mussel shell. And from that, we can count the annual rings and figure out the age of the mussel, and we hope to be able to cross date these. So we hope to be able to date one set of mussel shell rings against another mussel, and do that for probably 100 or 200 from this collection that we have, and from that build a long-term chronology. So dating one piece of this mussel's life against another mussel's life, and take that further back and be able to look at this continuous chronology of the life of these mussels. And then from that we can tell their age, how old they were, when these mussel shells died, and we hope to be able to drill into them and look at their chemistry as well. So if we can look at before the Clean Water Act and after the Clean Water Act, we can see if that law actually had a change in the chemistry in the Wabash River. So if you're interested in these, you can actually just pick these up and count those ridges. So this one's a pretty good example. You can see these very distinctive ridges on here, and if you just count those, you could figure out roughly how old this mussel was. So it depends upon the species and how they grow. This one's very distinctive. There's other smaller ridges on top of that, and that takes quite a bit of work under the microscope to figure out what's an annual band that's continuous throughout the shell versus just a small ridge. But on this one, just guessing, it might be like 10 or 11 years in age. Interestingly, these we worked on this during a tree ring class project. This is back in 2019. And the students, we only had enough time to surface up 3 of these samples. The youngest that we found was actually 27 years old, and the oldest was 54 years old. So we can look at that age. So some question about just looking at the surface, about how accurate you can be versus doing this extra work, we could tell a lot more from them. But if you're interested in the mussels, you can pick them up, count the ridges, and most likely you're looking at annual rings on them. So right now they're in my lab, and we're still working through the analysis. We hope to have them worked up later. We do have display cabinets throughout the science building at Indiana State University, and we can have some of those up in the cabinets later. Hopefully during the Smithsonian traveling exhibit, we can have some of these mussels on display at that time. I have a new graduate student, Hillary Howard, who is actually looking to work up this chronology. As I mentioned, we started this as a class project, and part of our process was actually figuring out just how to surface these. So this was a new analysis for us. I've never done this before. I've read about it. So we spent a lot of time kind of figuring out that analysis. Now that we have that procedure down, we can spend more time on it, and we have probably about 200 mussel shells collected. So Hillary, for her master's thesis, will be working through all of these mussel shells, trying to get them dated against each other, and probably look into that chemistry component as well. We can do micro drilling on there with a Dremel, collect that dust, and put it through an inductively coupled plasma mass spectrometer, and be able to look at the chemistry. So what heavy metals are they picking up through time? So there'll be hopefully quite a lot more work done with this through Hillary's master's thesis, and she's just starting that process now. And it's normally a 2-year process.